All right, this is chapter 16, which is the autonomic nervous system. And I can't draw as much on this presentation because my computer is out of batteries and it won't let me go into tablet mode when it is, which makes drawing on the screen really difficult. So I'm going to keep my eye on the battery and shift over when I can. But this initial part is not going to have as many annotations as I usually make, so just heads up. So in the last video presentation, we talked about motor pathways and sensory pathways. And in that one, especially in the motor division, we reserved discussion of smooth muscle, glands, cardiac muscle, and fat, those effectors. We had reserved those for discussing the autonomic nervous system specifically instead of talking about their motor pathways in the pathways chapter. And that's for a good reason. And it's because the autonomic nervous system is divided up differently than the rest of the pathways that we discussed. There's some more details and nuances to it, and so it deserves its own chapter and also its own presentation separate from the rest of it. So we're going to talk about these effectors and what they tend to do in response to different kinds of autonomic stimulation. We will also discuss their pathways and the kinds of neurons that make up the autonomic pathways, because the autonomic nervous system is divided into two kind of halves that work together, excuse me, to regulate your autonomic and involuntary homeostatic functions. And those two groups of neurons release different neurotransmitters. So we'll talk about that. We'll also discuss the location of the ganglia for the autonomic nervous system. And hint, they are outside of the central nervous system. They are positioned all over the place, but they're not like the dorsal root ganglion, for example. They have other ganglia that are accessed primarily by the Rami communicantes. So let's get into it. So when we say visceral motor pathways, that's a general term meaning anything that's not somatic and therefore skeletal muscle. So we're talking about smooth muscle, cardiac muscle, glands, specifically exocrine glands, and adipose tissue. And with respect to exocrine glands, there is one exception to the exocrine thing, and that is the adrenal medulla, which is an endocrine gland, does get involved in the fight or flight response, but that's really the only exception. So these smooth muscle examples are perhaps the most powerful in addition to the cardiac, because this is how we're going to do things like change our heart rate and contractility, so how hard the heart is beating, in response to preparing to fight or flee, or slowing down at rest, or even as we're dropping into sleep. And airway and blood vessels specifically are going to change a lot depending on your activity as well. And this can really change the way blood is distributed in your body and how much oxygen it gets, depending on the situation. So we'll discuss that also. So here we have an orc holding a very upset child. I don't have children, but I personally wouldn't do that to a kid and then take a picture of them. That seems inhumane. I understand that people do things because it's funny, but like, yikes. Regardless, though, this is a pretty accurate facial expression for the sympathetic nervous system response, which is our quote unquote fight or flight response. So this increases activity when we are exposed to stressors. And remember, a stressor definitionally for physiology is anything that disrupts our homeostasis. But it's also important to note that, like, even though we call the sympathetic division fight or flight, which tends to make people think of it as only an emergency portion of the nervous system, that's not actually true. So there's other situations which are not emergent that the sympathetic division is active and required for, like exercise, for example. So I would urge you to not think of it as only for running away from or punching a zombie, for example, but also involved in more day-to-day activities, especially in cooperation with the parasympathetic division. So the sympathetic division is very draining because it's energy consuming. And this is actually one of the reasons why people that suffer from PTSD or are living in war zones, for example, where their average stress level is high, tend to have their energy needs not met, and they tend to suffer pathological consequences of a prolonged stress response because this is for brief periods of activity, really. It's not designed to be present long term. We'll talk more about the general adaptation syndrome, which is a characterized and very well understood stress response in vertebrates and mammals, which we are both. So just save that for later. It's coming down the pike later. So the parasympathetic division is energy conserving, which means that it's going to prioritize not using your onboard body fuel as much as possible, and not overusing it either. So maintains your resting metabolism, which means that, one of the reasons our lifespan is what it is because we're not spending all of our time under extreme stress. We are primarily in a state of resting metabolism, so active but not hyperactive. So the parasympathetic division we kind of refer to as the rest and digest division. So again, fight or flight and rest and digest are handy for memory purposes, but I would urge you to not pigeonhole either division. They do work in concert with each other, and we'll talk about that when we discuss dual innervation. Right now. So dual innervation means an organ or effector is innervated, meaning supplied with neural input by both autonomic nervous system divisions. So this allows very fine control for things like heart rate, among other things, and also other stuff like, for example, your pupillary diameter. So there's two muscles in your iris. One is the constrictor and one is the dilator. They push and pull against each other to determine what your pupillary diameter ends up being. The sympathetic nervous system innervates the dilator, and the parasympathetic nervous system innervates the constrictor. So there are exceptions to dual innervation. For example, only one division innervates sweat glands, adipose tissue, lacrimal glands, and blood vessels, and that is going to be the sympathetic nervous system. Interestingly, one of the most extreme treatments for hyperhidrosis, which is excessive sweating, and this is like medical hyperhidrosis, so not like, ew, I'm sweating a bunch, but like, I won't give people handshakes because my hands are always wet and people think that is gross. Like a problem. Since the sympathetic nervous system is the only division innervating the sweat glands, you can have what's called a sympathectomy on your brachial plexus, or excuse me, not your brachial plexus, a sympathectomy that goes to the sympathetic nerves that supply your sweat glands in your arm and hands, and you can have them clipped so that your arms and hands no longer receive innervation from the sympathetic division, and that effectively deadens your sweat glands in your hands and solves your hyperhidrosis problem. However, one consequence of that would be it can make it difficult to lose fat in your arms, and depending on how the nerve is clipped, it can interrupt the ability for your blood vessels in your arms to dilate and constrict. So that's why I characterize it as an extreme option. There are other options that are not quite so bad. So cooperation of divisions is required for reproductive system function, among other things. So for example, we'll talk about this next term as well when we talk about the reproductive system. The parts of the sexual response that are basically like facilitating gametes meeting, so promoting the likelihood of an egg meeting a sperm. On the male side of things, uh, this is going to be emission, which is movement of sperm into the ejaculatory duct, and secretion of prostate products and seminal vesicle products to combine to create actual semen. Semen's not just sperm. That part is called emission, which is we're preparing the semen for delivery. And that part is regulated by the parasympathetic nervous system. The sympathetic nervous system is involved in the processes of ejaculation, so excitation to an extent that the semen is ejaculated and therefore has a much better chance of meeting an egg. And the same kind of situation is responsible for the female side of things as well. So Bartholin's glands, which are glands by the vagina, those are driven by parasympathetic innervation, and the sympathetic response is involved in female orgasm and, in some cases, female ejaculation. So that's what I mean when I say there's kind of a handshake between the two divisions to accomplish certain physiological goals. Those are two examples. Okay, so important vocabulary. What words am I going to use, and what do those words mean? So when you're consuming new information, especially audiovisual information and not reading, the more times I say a word that's not, familiar to you, the more likely you are to kind of get freaked out or tune out because you're like, wow, this is just too many new words and I don't know what any of them mean. And therefore, it makes the information being presented very difficult to understand. So, in the interest of avoiding that problem, I'm going to define all of these words. And I think actually what I'm going to do for this, I can't really draw, but I can use the text tool. So I'm just going to type these out. So autonomic nuclei are, regions of gray matter, cell bodies, unmyelin, well, let's do a carriage return there, unmyelinated axons and glial cells in the CNS, and primarily in the brain and brainstem, so the hypothalamus and the so the hypothalamus obviously is the diencephalon, and then the brainstem, the pons, and the medulla both have centers that, believe it or not, some of them correspond to cranial nerves. So imagine that. So these are in the CNS, and they're surrounded by white matter because that is the definition of nuclei, but these are autonomic ones. So visceral effectors, not affectors. Quick grammar lesson. If you're trying to figure out whether to say effect or affect, effect is a response to a particular thing. So thing A has an effect on thing B. Affect means either you're come across, so if someone says like, you have a flat affect, it means you don't speak with much emotion or tonality and you don't really make facial expressions. And then affected by means something applies an affect to something else or changes its affect. So just, because online communication is like how we get jobs these days, I want to sort of spare you from making a grammar mistake that might interrupt your possibility of getting hired or some such. And my walkie-talkies just went off for no reason, which is scary. So I have no idea who that is. I'm going to pause this and go figure out what's going on with that. I'll be right back. Okay, I'm back. Now that I've finished having the living daylights scared out of me, I realized my walkie-talkies are not haunted. I was just picking up somebody else's nearby signal. So that's a relief. Hopefully it wasn't hunters though, because this is a no hunting property. So if I have poachers, I'm going to go have to deal with that. All right, back to vocab. So a preganglionic versus postganglionic. This is a distinction that's specific to the autonomic nervous system. So you're going to hear me say these words a lot. So in each pathway, you can see that there are, after the initial nuclei in spinal cord or in the brainstem, depending on your division, there are autonomic ganglia. So the neuron that is going to the ganglion and synapses with the postganglionic neuron. So we refer to these neurons based on their proximity to the ganglion and if they're coming to or going from it. So a corollary to that is the postganglionic fiber. This should say post. So that's an error I will need to fix. So let's just go ahead and cross that out. Oops, nope, not like that. So we're going to go ahead and let's white that out actually. And I'm just going to type over it. Nice and easy. Oh, good Lord. Come on. There we go. Sorry. So postganglionic neurons have cell bodies in autonomic ganglia and extend from those ganglia to visceral effectors. And then autonomic ganglia I've already described, so ganglionic neurons, postganglionic fibers. Neurons and fibers we're using interchangeably here, so fiber just refers to the fibrous nature of axons, right? Okay, so that's the vocab. Now let's move on, having established that, to the rest of the autonomic nervous system. So looking at this picture here, we're just going to go ahead and look at the titles first and some positional differences. So the sympathetic division, fight or flight, activity, energy consuming, is also called the thoracolumbar division. And you can see that that's true because it's this portion of the spinal cord that gives rise to the sympathetic fibers, specifically the preganglionic fibers going to the ganglia, which then connect to the postganglionic fibers, which end up at the effectors. So the origination point here is the thoraco and lumbar spine. By contrast, the parasympathetic division is also called the craniosacral division, and this includes the brainstem, so the pons and the medulla, and also the spinal cord. So unsurprisingly, we've got cranial nerves here. So oculomotor, facial, glossopharyngeal, vagus, which is a powerhouse of a nerve, and then pelvic nerves innervating the pelvic viscera. So craniosacral, thoraco, lumbar. So they're spatially separate as well as functionally separate. So I'm just going to get all my text on the screen here because I don't like this little sliding over thing. So if we're comparing these two, I already mentioned this and we saw what it looks like. So somatic activity, somatic here means body. So this is going to increase somatic activity and this is going to decrease. And by activity, we mean excitability and also metabolism, so metabolic processes. Sympathetic is energy expending, so it's going to be your friend when you need to do a short burst of intense exertion. It's also involved in the alarm reaction and activated by mental stress, and physical activity, so exercise. So if I were to go charging up the hill to the north field, I would be activating my sympathetic nervous system, but not because I'm upset, it's just because I am going up a steep hill. So parasympathetic nervous system decreases somatic energy, so it's designed to conserve your energy. And this is active for resting conditions where you should be most of the time, although we live in a very scary, intense world right now, so it's understandable if you're not living in resting territory most of the time. I see you and I relate hard. Everybody's struggling right now. So if you're watching this video in the future, it is currently 2022, and things are not all that chill. So hopefully it's going to get better. So these are going to be responsible for things like SLUD, which is just an acronym that you can use to remember this, salivation, lacrimation, so saliva production, tear production, urination, digestion, and defecation, among other things. So side note, enteric nervous system refers to a specific set of neurons that are located in the muscular walls of the GI tract. And these are actually more independent than you might realize. So they are innervated, excuse me, they are innervated by the autonomic nervous system, but the action of the ANS only serves to adjust the enteric nervous system. It doesn't control it or dominate it. So the enteric nervous system kind of does its own thing, and then it increases its activity or decreases its activity in response to autonomic innervation, but it's very capable of acting on its own. So we'll talk more about that in Bio 242 when we get to the digestive system specifically. So all autonomic pathways have two neurons after their origination point. So we're going to ignore the hypothalamic neurons for right now. So the soma of the preganglionic neuron is in the central nervous system, and its axon extends to an autonomic ganglion. And then a ganglionic, also sometimes called postganglionic neuron, has its soma in the ganglion, and its axon, however long or short, and there are differences that we'll discuss, that's going to extend from the ganglion to the visceral effector. So when you hear me say preganglionic and postganglionic, remember that I'm referring to that. So now we're going to get into some pathway stuff. So the preganglionic neuron somas, which are the visceral motor neuron somas, those are going to be in the lateral horns of the thoracolumbar region of the spinal cord. So you might remember when I was doing the spinal cord sectional model tour, and the vertebra around the spinal cord section made it very obvious that that particular section was located in the cervical spine, and yet there was a pronounced lateral horn, and I mentioned that that was wrong. This is what I was referring to. So the lateral horn is in the thoracolumbar region because it corresponds to sympathetic control, which is in the thoracolumbar region. So that's where the lateral horn is, and not up here in the cervical spine, because that's not associated with the sympathetic division. So these are going to synapse in sympathetic ganglia and then extend to their various effectors, which are pictured there. So let's have a little look-see. Sorry, my remaining dog has been very needy since his buddy passed, and he's trying to get on my lap. Even though there's a computer on my lap, you can't get on my lap. Just lay down next to me. You'll be fine, I promise. You're not going to wither away from no attention. English bulldogs are really needy. Okay, Bubba. All right. So as you can see here, we've got our dorsal ramus and our ventral ramus, which are going to branch to supply the dorsal surface of the back and the ventrolateral surface of the front. And then we have the two Rami communicantes, that's plural, and that's because are two of them, even though your model in the lab does not reflect that. It's because they're both sharing an epineurial covering, so you can't see the difference between the two. So we can see we have a white and a gray one. So the white ramus is sympathetic, and this is specifically because preganglionic axons are myelinated, and myelin appears white in section. So that's why it's called the white ramus. And then the gray ramus is called that because those fibers are unmyelinated and therefore are gray instead of white. After the sympathetic ganglion, which we will discuss, they combine to form a sympathetic nerve, which contains both preganglionic and postganglionic fibers innervating stuff in the thoracic cavity. So we'll discuss that synaptic arrangement in a little bit, but that's why those structures that you learned in lab exist. So the Rami communicantes are providing autonomic innervation via the sympathetic ganglion and the Rami communicantes to act on visceral organs in the thoracic and abdominopelvic cavities. And you can divide these up as well. So postganglionic fibers are going to go to smooth muscles because the ganglion in this case is the sympathetic ganglion. And then preganglionic fibers are going to innervate abdominopelvic viscera. So that's kind of the deal there. So we've got sympathetic ganglia, 3 categories. So this should look familiar from your model, and then this maybe less so. This is kind of a weird representation of them, but hey, it is what it is. So these are the sympathetic chain ganglia, or it's also called the sympathetic trunk. So they are a series of ganglia that hug the body of your thoracic and lumbar spine and are connected to each other up and down, as well as to the rest of the spinal nerves via the Rami communicantes. So here's what they look like in situ, where you can see them running down the thorax and upper abdomen and then giving rise to other nerves. So we call these the sympathetic chain or sympathetic trunk ganglia. There's also collateral ganglia, which are further out, and that weird exception I mentioned earlier, the suprarenal, also called the adrenal medullae. Medulla just means the inner portion of a gland or structure. So sometimes the inner regions of an organ are different in function and structure from the outer regions, but they're all kind of cobbled together into one gland. The adrenal gland is one of those, and the medulla specifically has an involvement in the sympathetic nervous system, so I'll explain that later. All right, so let's have a look-see at the wiring up of sympathetic chain ganglia specifically. So we can see we have a preganglionic neuron with its cell body in the lateral gray horn of the spinal cord. That preganglionic neuron is going to extend out. It's going to join up with the spinal nerve here, so briefly becoming a mixed nerve, and then it goes downstairs to the nearest sympathetic ganglion, and that's where it synapses. So this is going to be via the white ramus because this is a myelinated axon. And then after that synapse in the ganglion, it's going to innervate visceral organs in the thoracic cavity. So forming things like the sympathetic cardiac nerve, for example. So the other option is we have cell body, again, in the lateral horn. We have ganglionic neuron synapsing here in the sympathetic ganglion. And then instead of going straight out, it does a little loop-de-loop. And in this case, since the loop-de-loop is being achieved by the postganglionic fiber, not pre, it is unmyelinated and therefore traveling in the gray ramus instead. So one detail here, which is important to know, is that in both divisions of the autonomic nervous system, preganglionic fibers are myelinated And postganglionic fibers are often or almost entirely not myelinated. So there's that division as far as myelination between the two, and we'll kind of build up that concept as we go, because what we're going to do at the end is do a nice compare-contrast between the sympathetic and parasympathetic nervous systems, stuff they have in common and stuff that's different. So collateral ganglia, remember I said we're outside, and so in this case, the preganglionic cell body is still in the lateral horn, and the preganglionic fiber that extends from it is still myelinated, but instead it just completely skips the ganglion. It's like, I'm going to go right on out here and synapse on collateral ganglia, which are positioned all over the place in the abdominopelvic cavity. So they don't even involve synapsing in the sympathetic ganglion at all. And that's just fine. The collateral ganglion positions them much closer to the organs, which helps to speed up the rate at which the action potential travels on its way to the organs. Helps it get there faster since the postganglionic fiber is unmyelinated. It makes sense for the preganglionic fast guy to be longer in this case, and that way the unmyelinated axon carrying a slower action potential doesn't have as far to go. So this is still going to travel via the white ramus because again, it's myelinated. And then we've got postganglionic fibers innervating all kinds of organs. Now notice nerves in the sympathetic nervous system containing preganglionic fibers have this fun word associated with them, and that word is splanchnic, which is super fun to say. I will give you the etymology or the breakdown of it because it's interesting, and it might help that weird word stick better in your minds. So we've got splanchnikos. I feel like there's an H in there somewhere. I'm not a Greek scholar, sorry. But splanchnikos is Greek for entrails. So entrails are another way of talking about your abdominal viscera. So the reason that gets a specific word in Greek is for a variety of reasons, but ancient Greek religions used an abominably cruel practice called entrails reading, where they would basically take a bull, usually because bulls were higher priority livestock animals. So sacrificing one was like kind of a big deal because by getting rid of a bull, you're also removing any potential he would have to help you produce more cows, right? So if you're trying to like impress the gods, you want to give up something that actually matters to you, and that would be a bull in that case for those reasons. But you bring a bull and you're like, wow, bummer to lose this bull. Hope Apollo is not mad at me. And here's a bowl for it. And then they would slice open the animal's guts, and a priest or priestess would read how the guts spilled out and kind of read it like tea leaves. But of course, this is incredibly inhumane, so yikes. But that's where the word splanchnikos comes from. And the reason that we're using it here for splanchnic is that, of course, it's innervating your abdominal viscera, so your guts. So entrails equals guts, basically. I try to give little doses of medical and etymology history here and there because context is what our brains need to help new information stay in and not lose it. And so maximum context is going to help you remember better because a story about Greek entrails reading is more likely to stick in your brain because it's narrative and it's remarkable than just, hey, this is a splanchnic nerve. Don't worry about what it means. It's just a nerve, right? So that's what the deal is with collateral ganglia. So preganglionic fibers enter to splanchnic nerves, which are still myelinated, and then pass through to collateral ganglia. And as I said previously, the ganglionic fibers, which are shorter and unmyelinated, are going to innervate effectors in the abdominopelvic cavity. Okay, so let's talk about our weird exception here. The inner core of the adrenal glands, so not the outer part, that's regular endocrine stuff, but the inner part is kind of special. So interestingly, and you can see it really well in this picture, so we have the two adrenal medullae pictured here. So this outer stuff is the cortex, and then the pink part in the middle is the medulla. Again, cortex means edge part, medulla means middle in part. So again, we have a preganglionic fiber passing through the sympathetic chain ganglion, unsynapsed, and traveling past the collateral ganglia and going all the way out to the suprarenal medullae, for which the postganglionic fiber is vanishingly short. And in fact, they don't have axons at all. So they are weird neurons that are called anaxonic, meaning they don't have an axon. And they just sit in there and wait for a signal from the sympathetic nervous system. And when that happens, they just dump epinephrine and norepinephrine into your blood. So epinephrine and norepinephrine are basically sympathetic nervous system alarm response superchargers. They do all kinds of things, and the benefit of having this wiring system is myelinated axons carry action potentials very, very quickly. And then additionally, in the adrenal medulla here, in addition to these weirdo neuroendocrine cells with no axon, which are technically still neurons, they have cell surface markers that are neuron specific, even though they're not quite the right shape that we would think of for neurons. So these guys are going to, in response to this rapidly deployed action potential, dump epinephrine and norepinephrine immediately into very large blood vessels that are right next to them in the center of the adrenal gland. And that helps to get these neurotransmitters out quickly and in large quantities during a fight or flight response. So basically, these are going to be energy consuming primarily. So these chemicals increase your neural activity and alertness and awareness. They're also going to cause a change in energy usage to shift it towards immediate energy sources and glucose sparing effects, and all kinds of other stuff as well. So the Rami communicantes, the white ramus is myelinated preganglionic fibers, of course, because white equals myelinated. And these are from visual observations of gross anatomy, by the way. And then the gray ramus are unmyelinated, postganglionic fibers. So in your lab, they were these little guys that are going to hop over the anterior part of this transverse process of this cervical vertebra. How do I know? It's got 3 holes, and if you could see up here, it has a bifid spinous process. So the lateral horn should not be here. This is an oversight. However, the Rami communicantes in this model were contained in this epineurial sheath and not separated out. So it was confusing to students. They're like, why is it plural if there's only one thing? And it's because there's 2 things in there, but the model didn't bother to make that clear. So white rami, because of the sympathetic activation, are located along spinal segments of thoracolumbar region only. Gray rami also contain other stuff, so they're located along each segment of the entire spinal cord, including C-spine, the rest of the L-spine, and the sacral spine. So how does this information or activation get out? So effectors in the head or neck are served by the sympathetic chain. And if you look at this part of the image, what you can see is that the ganglia extend up, even though they're not directly connected to the cervical spine. There are some up here, so basically the myelinated preganglionic fibers just climb this little rope and then synapse at the appropriate level to innervate head things. Thoracic cavity viscera served by sympathetic nerves, abdominal cavity viscera, guts served by splanchnic nerves, and effectors in the body wall and limbs are served by spinal cords, or spinal nerves, excuse me. So this is going to be basically dorsal ventral ramus stuff, but very few sympathetic fibers in those spinal nerves because the primary purpose of the dorsal and ventral horns are, of course, to do somatic stuff. So sympathetic neurons are the minority here, although they do exist. So let's talk about divergence. So we're going to circle on back to our discussion from previously of neural pools or circuits. And this is a picture, a basic diagram of a diverging circuit. So this means that one preganglionic fiber can synapse on up to 25, in some cases, ganglionic neurons. So one of the benefits of divergent pathways is that they're really good for rapidly deploying a necessary homeostatic change. And I can't think of a better, use for rapid deployment than a fight or flight response, right? So if you need to fight or flee to survive, or try to anyway, having a slow message system neurally would not make any sense. We really want a fast one. So we want to get that information out and the changes made in a jiffy, and divergence accomplishes that goal. So what does sympathetic innervation do? It dilates your airway and it increases your respiratory activity. It increases your blood pressure and heart rate, and these two things are linked. So the faster your heart is pumping under normal circumstances, the higher your blood pressure will be because there's less of a relaxation period between ventricular ejections, which puts more blood into your arterial side of your circulation. It's going to send blood to your brain to increase mental alertness, along with the epinephrine and norepinephrine helping that. It dilates your airways, as I mentioned, which falls under respiratory activity, really. It increases sweat gland activity and muscle tone. Other things that it does, dilates your pupils, for example. It also increases metabolism. So here we mean, aerobic metabolism, and in some cases, anaerobic. But basically, we're just going to upregulate the rate at which ATP is used, or we're going to make ATP from stuff that's not ATP by recruiting adipose tissue primarily. So we're going to basically take the triglycerides out of adipose tissue and stick them in the Krebs cycle and mine them for potential energy to make ATP. And then in skeletal muscle and the liver, we're going to do something called glycogenolysis. So glycogen is a big branchy molecule that is just basically a bunch of glucoses glued together. And it's a good way to store glucose for an emergency. So glycogenolysis means we're going to take that glycogen and start hacking glucoses off of it because that's what it's there for. It's a reserve glucose storage thingy. It's going to decrease GI tract and urinary tract activity. And this is all up and down the tract as well. So specifically, the formation of new urine. So in a fight or flight response, and someone always asks this in class during actual lecture, so I'll just address it here as well. But regardless, essentially what's going to happen is that it's not really important to digest a sandwich or to, make new urine if you are busy trying to survive by running away or acting aggressively. So that stuff just gets put on pause. And the primary mechanism for that is just blood is diverted away from these things and to your arms and legs. And that helps to damp down their activity as well as some degree of neural suppression. But it is still possible to pee yourself in fear. So voiding your bladder as a fear response is different from dampening all urinary tract activity. So the muscular actions that allow micturition, which is urination, are a little bit separate from this. Okay, so neurotransmitters and sympathetic function. Here we have a nice compare-contrast, which we're going to build up. So sympathetic nervous system-wise, the preganglionic neurons are cholinergic. Remember, that means they release acetylcholine. And this is an excitatory synapse, meaning that acetylcholine release by the preganglionic neuron onto the postganglionic neuron is going to excite this neuron and cause it to send an action potential to somewhere. So in this case, into the bloodstream via the adrenal medulla, and also to the target organs and tissues via any number of ganglia. So let's talk about the postganglionic neurons. Most of them are adrenergic, releasing primarily norepinephrine. There are a few cholinergic ones, but not very many, and there are a few nitroxidergic ones also, not very many. So nitroxidergic neurons are ones that release nitric oxide, which is a gas, but also a neurotransmitter. We call it a gasotransmitter. And these are also rare. These ones are primarily about dilating blood vessels in appropriate regions because nitric oxide is a very potent vasodilator. Okay, so here's an interesting thing about ganglionic neurons. They don't have axon terminals in the way that you're used to seeing. So that little knobby end I usually draw, not present here. Instead, what they have are called varicosities, which is where there are these little bulges, all of them with vesicles and all of them with the voltage-gated calcium channels that permit vesicular exocytosis, and those spread out via collateral branches that are full of varicosities to wrap around whatever's being innervated. And that is a really effective way to release lots and lots of neurotransmitter onto something almost all at once and very quickly and efficiently, hence this weirdo arrangement. So most of these are norepinephrine, so adrenergic. So what happens to the norepinephrine after it, after it gets released? Well, of course, it's going to bind to receptors on the effector cells and change their metabolism to serve some sympathetic related purpose. It can also be reabsorbed into ganglionic fibers to be reused, which is handy because we don't want to have to make fresh norepinephrine every time. That would take too long. Or it's also broken down by MAO, monoamine oxidase. So monoamine neurotransmitters and hormones are ones that are derived from a single amino acid. So typically tryptophan is the most common one, but there are some others. Tyrosine, for example, is this guy. So monoamine oxidase is an enzyme that basically oxidizes any monoamine neurotransmitter or hormone by breaking it down. So if you take an MAOI, an monoamine oxidase inhibitor for depression, serotonin is also a monoamine. So the effect of taking an MAOI is to inhibit this activity so that more serotonin ends up in the synapse, which increases synaptic action that is serotoninergic. So if you're taking an MAOI, one consequence of that might be that epinephrine and norepinephrine have more effect on you while you're taking that medication. So when it diffuses out of the synapse, we don't want the norepinephrine to wander away and cause inappropriate activation in a place where it's not supposed to happen. So if that happens, basically we have this little roving policeman that makes sure that it doesn't diffuse out of the synapse and it remains in the appropriate location. So this is called catechol O-methyltransferase, again, another enzyme, and just it basically walks up to the monoamine and says, no, you do not get to function, and it chemically changes it so it can't. So how long does a sympathetic event last after one issuance of a sympathetic signal? So postsynaptic effects last for several seconds, so not very long. And again, we don't want rogue epinephrine and norepinephrine to muddle our workings when we no longer need to be in fight or flight response. So we've got catechol O-methyltransferase, or MAO, monoamine oxidase, to basically just say, hey, nope, you're not allowed out of the synapse, get back here, you have to be in this one place. Interestingly though, neither catechol O-methyltransferase or monoamine oxidase are in the blood to break down the epinephrine and norepinephrine from the adrenal medullae. So once those chemicals are released into your bloodstream, the long-reaching effects last longer because we have it in circulation and those two things are not around in the circulation to break it down. So this allows circulating epinephrine and norepinephrine to be elevated for 30 seconds or more, as opposed to the postsynaptic kind. So all this corresponds to affecting effectors, which is a fun thing to say, for a duration of several minutes. So that's why when you get startled or scared or become extremely angry, it takes you a little while to sort of wind down from that response because you still have circulating neurohormones that are activating, bopping around in your blood, and they need time to get cleared. Okay, so in this next portion, we're going to look at specific neurotransmitter action. And I know this illustration is a little bit intimidating because it has some unfamiliar words, but it's not actually that bad. And if you learn this well this term, you're going to have much less of a hard time understanding the effects of hormones binding to their receptors. So let's get this out of the way early. Oops. So what this is showing you is an indirect effect where a neurotransmitter binds to a receptor, but unlike a cholinergic receptor, this receptor is not also a channel, and it's associated with the G protein. So G protein is asleep until the receptor binds to the neurotransmitter, in this case epinephrine or norepinephrine, and that wakes the G protein up, which wakes up adenylate cyclase, which is a enzyme that takes a phosphate off of ATP and then another one. And not only does it do that, so that makes it regular AMP, but cyclase means it turns it into a cyclic molecule. So not only is it AMP now after 2 phosphate groups are removed, but it's also now a circle or ring shape, whereas regular AMP isn't. So cyclic AMP is a important form of adenosine monophosphate, and that basically does a bunch of things. It's a really powerful second messenger, but in summary, it just activates enzymes that change cell metabolism and activity. And how exactly it does that or what exactly it does is going to really depend on what cell we're looking at. So there's no like one size fits all what this is, other than this vague statement. Another thing it can do is open ion channels to help the cell in question depolarize if necessary. So alpha and beta receptors come in 2 flavors. Remember, they're all adrenergic, but there's different kinds of them. So alpha receptors can be divided into alpha 1 and alpha 2. So alpha 1 are going to cause calcium ion release from the endoplasmic reticulum. So the importance of calcium should be pretty built up for you by now. We need it for muscular contraction in all types of muscle, and we also need it for neurotransmitter release. So this contains a function that means it's usually excitatory on smooth muscle because that calcium is going to bind and complex with calmodulin, and it's going to activate excitation and contraction. This also constricts the sphincters of the GI tract and urinary bladder. Alpha 2 receptors actually lower cyclic AMP instead of activating it. And in some cases, this is activatory, but usually it's inhibitory. So it basically tells key enzymes to stop. And this is usually inhibitory at neuromuscular and neuroglandular junctions. So the reason, for example, that you might get dry mouth if you are scared or upset is because your salivary glands are being inhibited by alpha 2 receptors, for example. Now, beta receptors come in 3 varieties. So their activation in general is going to increase cyclic AMP, which causes a change in the effector, but that change is not one size fits all, of course. So beta 1 receptor activation increases metabolism of skeletal muscle, which is good if you're trying to run away, and it also increases heart rate, so speed, and the force of contraction. Beta 2 receptors relax smooth muscle in the airways, so the tubes that let you get air, they open up so you can get more air, and blood vessels both to the heart and to skeletal muscle, which makes tons of sense, right? Your heart is what sends blood to your skeletal muscle, and your heart is also a muscle, so both of them are going to need lots of oxygen. And then beta 3 receptors are for lipolysis, so they're going to release fatty acids from triglycerides into the blood so that tissues can use them to make ATP. So if you are familiar with the drug that is a beta blocker, which is used to treat both stress and anxiety, but also like situational anxiety, but also heart medication, these are basically going to suppress beta receptor activation, specifically as it relates to the heart. So by suppressing an aspect of your sympathetic nervous system, which tends to be self-reinforcing, so the feeling of a pounding heart to us means something's wrong. If you can decrease that, it will relax you somewhat, and that's what those drugs are designed to do. So the cholinergic fibers are very few in number, but they do exist. So they are in both sweat glands and blood vessels that go to skeletal muscle. And so that's going to be things like the femoral artery. So we're getting our legs ready to run, right? And so we want to pump more blood there. So acetylcholine is going to dilate those in that case and also increase sweat gland activity. And then we have these super fun but very rare nitroxidergic ganglionic fibers. So the effect of these is going to be, as I mentioned, very vasodilatory. So they relax the smooth muscle in blood vessel walls, and that causes the blood vessel to dilate, and the dilation means that more blood can get through to the place where it's needed. So we're going to relax those smooth muscles and open up this blood vessel so that whatever tissue is downstream of the blood vessel is going to get plenty of blood. Okay, so I'm going to stop there because this is long enough. So we're going to do the parasympathetic division in the next video, but in the interest of timeliness, I'm going to just go ahead and get the sympathetic section uploaded, and then I'll resume recording so that we've got a constant stream of uploads. I think that's the best choice here. So as always, thank you for your attention, and I will see you in the next video.